Okay, let's get started, guys. So I have a few announcements for you before we get started. Hi. It's funny, at the beginning of the semester, everyone is on their own, and then you see these small groups starting to form, and then the class gets louder and louder and louder. So biggest group formed is at the back right there. They're just missing Amir. I don't know where he is, but that's okay. What did I want to say? Okay, so we have 4 lectures left, including today. So in 1 hour and 15, that's 3 lectures left. We're going to cover population genetics. We're going to go into genomics, and then we'll do a review session or 2, depending on what you guys want. Okay. If you are free on the 16th, maybe we'll do 1. I'll see how it goes. So that's for that. We have a quiz tomorrow, which I'll be posting tomorrow at like 9 a.m. It will be on protein synthesis as well as lacopron, the first part. I'll leave it open for the whole day. I'm not going to be crazy about the timing, so you can use your books to do whatever you want. Just do the quiz. It's good practice for you. And then you'll have one last quiz the week after. And it will be on probably gene isolation manipulation. Okay, so the restriction enzymes and all that. Cool? Easy? Yep. It's just because Monday there was no class, so I had to remove the quiz from being done in the tutorial session, and then I moved it to online. So everyone will do it online. Okay. Lecture 19. So there was a problem with the recording for lecture 19. The file was corrupted. I tried to fix it. I think I got it today. So, but there's a part of it that's missing. So I'm going to try to edit that today and post it either by tonight or tomorrow. Hopefully it will work. If it doesn't work, I don't know. If I have some time, I'll record it. Otherwise, just go through the lecture slides. And yeah, that's it. For the people that are celebrating Passover, the exam is on the 19th. Now I'm going to give you a choice to do it at 9 a.m. or at 2 p.m. So what do you guys want? Sorry? 9? Okay, so you guys want to make me wake up early again? Okay, we'll do it at 9. That works for me. So I'll send you the place where you have to do the exam. It's going to be different from the one that's on the 23rd. So be ready for that. And other than that, well, I think that's pretty much it. Are you guys going to miss me in 2 weeks? Yeah.
You're saying about the 19th, that's the placement?
It's for people who are celebrating Passover during the week of the exam. So for those, I have to give an alternate date. Well, I don't really have to, but I'm doing it. And so for them, it will be 3 days before, 4 days before. Okay. This is the first time. I didn't even know that these alternate dates existed in my when I was studying. Otherwise, I would have used all of them. Anyways, so population genetics. It's a very simple topic, but it has a little bit of math. So follow what I'm saying here. Hopefully, you're going to be able to do the simple math. Obviously, since we're covering, there's a question about this on the exam. So try to understand what's happening here. Before I get into that, I forgot to ask you, do you have any questions for me or concerns? No? Okay, so let's get to it. So since the beginning of the semester, we've been talking about Mendelian genetics when we were analyzing the crosses, right? And we were looking at it as being, so let's, for example, take a cross like this, where you have 2 homozygous parents. Then it gives you a heterozygous offspring. We were always considering it as being each allele has an equal chance of being transferred, right? If it was like this, it would be much different when we were talking about like diseases. For example, if we're talking about the dominant allele, you should expect it most of the time in the population, but that does not happen like that. And this is because of a concept called the gene pool, right? So the gene pool is, you can imagine it as being a bag of alleles, and there's a frequency for each allele in that gene pool, right? And because of the way it is the gene pool is sorted, then you'll end up with different alleles at different frequencies. And so their inheritance is not into a 0.5 or 50%, assortment. It's more like 0.1, or depending on which allele we're talking about, okay? So I remember at the beginning of the semester, some students were asking me, they found this weird, like, how is it that we have a dominant allele, but it's still not the predominant phenotype in the population? Because if you have one allele and you get the disease, you'd expect most people to get that disease. Well, as I just said, that's because of the frequency of each alleles in a specific gene pool, right? And the gene pool could be considered, so it has a space and time kind of. So for example, you can consider the gene pool of the whole Canada, right? Or the gene pool of just Montreal, right? So depending what population you are studying and at what time, obviously. So population genetics is an interesting study for retracing, for example, our ancestors, how they looked like, what the diseases they have, because we can go backward in time by playing with these frequencies, let's say, okay? And we'll see how to do this today and hopefully next lecture. So, so this is what I just said, basically that disease-causing alleles, they occur usually very rarely in natural populations, and this is just due to these frequencies. We'll see how the better fit organism will survive while the sick one will die, and so this will change the gene pool, and this is why you see diseases that are very rare in the population. For example, celiac disease, the way I presented the allele was dominant, right? And it does happen in my family, but I'm pretty sure that not everyone here has celiac disease, right? And this is just an example of how these frequencies can play a role, okay? So we'll be learning about how to determine the frequency of these alleles, their relationship to the genotype. We'll also be looking at the effect of inbreeding. So inbreeding is when there's breeding within the same family, kind of. This is mostly considered for plants, but there are some situations where it happens even with humans, which is highly illegal. And this has an effect on the genotype frequency. So there are specific parts of the world, I don't want to get into the parts of the world, you can Google that by yourself, where there's a lot of inbreeding. And so you can research that and you'll see that those people have a lot of genetically inherited diseases. This is like a known fact, okay? So you can Google this. I'm not going to start mentioning who's doing what there because I don't want to get into trouble. Get Google in trouble, okay? And lastly, we'll be looking at selection, mutation, and migration. So how these selective forces have an effect on the gene pool and why do we have the diversity that we see today. The chapter covers a lot of stuff, okay? Single nucleotide polymorphism, microsatellites, and it goes on, okay? The only things that are relevant for us is this stuff here, okay? If you want to learn more because you're interested about population genetics, go for it. But exam material and course material, just the lectures will be sufficient. So let's take a look at an example where we have different frequencies based on the type of population, right, that you have. So there's another blood type called the MN blood type. So it's similar to the ABO blood type. It's also a codominant system, just that you have two alleles, the M, the N, instead of the A, B, and then the recessive allele. And so you can see that different populations have different frequencies of genotype. So we can look, for example, to, I don't know if this is like a, you have to be careful when you talk about this stuff. So I don't know if this word here is offensive, but they basically have different frequencies for the dominant allele compared to the recessive allele. And you can compare it to different populations and you'll see that they have different frequencies, okay? So this will tell you that, for example, in this group here, they will have a skewed blood type towards, you know, something. I don't know what the M blood type is relative to the ABO, but they will have an M blood type, let's say, okay? Now, as you can see, these frequencies change based on the group we're studying, right? And so without getting too political and saying too much weird stuff, but basically you can see how James Watson was thinking, right? When he said that potentially race affects intelligence, right? And so it's because he's basing it on stuff that he sees like this. It's just there's no actual research that was done to show if there's something or not. We can see that, you know, from where you come in the world, it affects your blood type, right? This is an example. There's also another example, which we'll see at the end of this lecture, which shows you about sickle cell anemia. And this is a disease that can be fatal. And you can see that it was developed in Africa, mostly as a protective mechanism to malaria, right? And so Africans have, tropical Africans, so sub-Saharan Africans have a lot of sickle cell anemia within the populations, but not because they are sick, because it's a protective mechanism that evolved against malaria, which is found in those tropical regions, okay? So there's like a two-edged sword here when you're talking about intelligence and race. You don't want to get into that politically speaking, and people are usually scared to dabble into that. So usually you just block it on the spot and no, there's nothing. But you can see why Watson was kind of thinking like this, because there is variation between groups. Just you don't link it to something like intelligence, you know. You have to play in the gray zone. So that's for that. And then the tests that you see, like 23andMe or Ancestry and all that, they use this kind of science to figure out from where you come, right? So they will take many loci for example, your blood type. Obviously, if you follow exactly this pattern here, you're an Eskimo, or I don't know, but you know, they follow this stuff and they can retrace you back to the oldest known civilizations, okay? Unfortunately, there's no Algerian all the time. I wish I could know what the hell was happening there. But my people are always forgotten in this kind of stuff because, you know, research is very weak. But anyways, so how do we calculate the frequency of alleles? So you probably heard of this. Oh, yep. What's up, Pedro? Right, so we'll see that right now, but when you have a gene pool that is closed without any like effect on it by selective forces, then what happens is that the gene pool will just keep renewing itself and the frequencies will remain the same, right? But then if one of them travels and leaves and goes to somewhere else where that blood type is not existent or very weak, then you have something called the founder effect, where that person will will be the founder of a new group that has changed, right? And so if you stay, for example, within the same gene pool, then you'll end up, if you're mating in that pool, you'll just keep the frequencies going. But if you go in a country, for example, where there's a lot of M blood type, right, or an area where there's a lot of blood type, you will be kind of eaten by that group because they have a lot of the M blood type, right? And so yours will be kind of hidden. You see what I mean? But it will change the frequencies. Does that make sense? Good. So the Hardy-Weinberg equilibrium. Who's seen this already? Shit. It's like the last few lectures and everyone saw it. Okay. So I guess this class is useless. Actually, no, it's not. So we'll see it in a more complicated way, okay? So the way we calculate the frequencies in a population, usually we are diploids, so we consider only two different possibilities. So if we have two alleles and we're calculating the frequency, let's say, for example, the alleles big A and small a, then the frequency of big A in the population, so the number of times A occurs in that population divided by the total population, that's P. And then if we're looking for the other allele, so this is in the case where we only have two alleles, okay? Then we'll be looking at Q, which is equal to the frequency of the small a in that population. So the number of people bearing the A divided by the total population, okay? Obviously, if you add them together, there are two frequencies. You can have only two possibilities. So if you add them together, you'll end up with 100%, right? So the frequency of A is 50, and this A is 50%, then you can see how if this one is 70, this one is 30, it will be like this, okay? So let's just take a small example here and then we'll get into more details. So if the frequencies of the genotypes in a population, you have, for example, 62% that are homozygous dominant, 16% that are heterozygous, and then 22% that are homozygous recessive, then you can calculate the frequency from these numbers, right? So the frequency of big A, which we, again, as I said, is P, that's equal to the frequency of this guy plus half of this guy. Is that simple to understand? Right? Okay. So that will give you 0.62 plus half of 0.16, which is 0.70. So you can go from these genotype frequencies to the allele frequency by doing this calculation. Okay. And then if you do the same thing for Q, which is the frequency of the small a, well, then you will take the frequency of the homozygous recessive plus half of the frequency of the heterozygous, add them together, that gives you 30% or 0.3. Okay? So that's that. If you add these, you'll end up with 1. If you add these, you'll end up with 1. Okay? So this, what I just showed you, is the Hardy-Weinberg equilibrium. It just describes the relationship between the alleles and the genotypes. And you'll see why is this relevant when you're studying population genetics. But basically, if we take these numbers from here and try to figure out some sort of formula from it, what it really looks like is, so if we consider the frequency of A as P, then the frequency of AA is P squared, right? Because it's the frequency to get one A from one parent and the frequency to get one A from the other parent. And so it's P times P, which is P squared. Q is the same thing, but for the other alleles, sorry. And then if you do the Punnett square for a cross, right, you'll end up with, in terms of genotypes, you'll end up with a 1 to 2 to 1, right? The 1 is the homozygous dominant, the 1 is homozygous recessive, and then you have the heterozygote, which happens in 2, right? So the frequency of A, big A, small a, is the frequency of A alone times the frequency of big A alone times the frequency of small a alone, so that's P times Q, and it happens 2 times in the Punnett square, so times 2. Easy? Okay. You add up all this together, what is it supposed to give you? Are you sure? Yeah, that's good. So this equilibrium here is like, we learn it, but in reality, it barely ever happens, okay? Maybe it will happen in some sort of isolated area for, let's say, a species, but not for humans, certainly not, because humans are not isolated. So this Hardy-Weinberg equilibrium assumes that you're dealing with a large population, that mating is random, and that there is no significant selection, migration, or mutation, because these will transform the, will actually affect the Hardy-Weinberg equilibrium, because, for example, migration will bring in new genotype, selection will select for one allele more than the other, and mutation, we'll see that in a second. So for humans, this does not happen. Why? Because actually mating is not random. We are more of the selective type. And so this equilibrium does not happen for us. You know, usually there's a few stereotypes about this. Tall guy, whatever. This is the exact thing. So this is an example of why the mating is not random. And there's no significant selection, migration. So migration, this is, everyone here is from a different country, probably, etc. But this Hardy-Weinberg equilibrium is used for crops, for example, so that you can produce better crops, because you can isolate those crops and then just generate more of them, right? And things like this. So what did I do here? Okay, that's the opposite scenario. If you start with the probabilities of each one, then you just have to calculate the genotypes. And the way you do that, if you have the probability, the frequency of just the allele A, big A, then it's this, if you want to find this genotype squared. So that's just the two ways of doing it, either starting from the genotype frequency to the allele, or the allele to the genotype. Okay, so one thing that I was just explaining here is that if the population is in Hardy-Weinberg equilibrium, that means the genotype, the allele frequency will remain constant within the population, because the population is in genetic equilibrium. You have the same number of big A's, same number of small A's, and then and same numbers of genotypes, so if they mate, they will produce the same frequencies. Cool? Pop quiz. Let's try this one here. Forget about the old type, blood type. Let's assume there's only 3 of them. Can you do this number here without looking at the next slide? Need a little help. Yep. D, E. Yeah. Okay. So if you're able to get it by yourself, that's good. I'll explain how to do it now. So if you have the frequency, so if you have the population is 1,000, right? So if you have the number of people that have the genotype AA, so you can find P squared, right? And if you find P squared, that's basically 160 divided by this number here, which gives you 0.16, and then you do the square root of that. That will give you P, and then you're able to do 1 minus that, right? And that will give you the frequency of the allele B. Or you can do it this way too, which is, let's see what they did here. So they did this divided by 1,000, so that gives you 0.3. Plus 1/2 of this one here. Okay, you can do it this way also, just by counting the alleles directly like this. So the frequency of this guy plus the frequency of half of the heterozygous, and it gives you, was this wrong though? No, this is good. Never mind. Okay. So with every math formula, there's a proof or a way to demonstrate that it works. These proofs are kind of important because you don't want to just follow this. Yep.
What was the first way that you said how to do it?
So to find, so you have here 160 of AA, right? So if you do that divided by this, it will give you the frequency of AA, right? But then you also need to add, wait, what am I doing here? Yeah, and then you need to add half of these guys, because there's an A here, right? And so if you add 0.16 plus 0.48, it gives you this. The other way that I was saying was, let me actually do it on paper first before I, 1 second guys, that's my math. Actually, doesn't really matter, just do it this way, but let me just try the other method. So 0.16, that's AA. And then AB, to PQ, which is equal to 0.48. Yeah, the other one requires some algebra. Just forget about the other one, okay? Just do it this way, it's easier. Just remember that when you're looking at the allele, okay, and at the genotype, that there's 2 genotypes that have the allele A. There's this one here, and there is this one here. And so you have to consider it as this one here, it's half of whatever it is, right? And then this one here is the full number, okay? Too complicated for nothing. Okay, So let's show this Hardy-Weinberg system, and also this proof here is going to show you that how you can use it to determine the mating between different genotypes and how often they will happen. So if we start with an arbitrary distribution of the genotypes, so just put any number that you want here as long as they end up being equal to 1. And then you find from there the frequencies of big A, small a, by doing 0.2, for example, for big A plus half of this one here. It will give you 0.3. So the frequency of big A is 0.3, the frequency of small a is 0.7. And then you can use that to determine the mating combinations, right? So you would put on both sides, kind of like not really a Punnett square, but you'll just put them on both sides, both parents, right? And then you can determine the percent of time that two different genotypes will mate together. For example, in the population, there's 20% of big A. And so what is the chance that they will mate together? Well, it's 20% times 20%, right? So it gives you 0.4. If you're looking at, and that's just basically 4% here. If you're looking at mating between big A, small a, and then the homozygous dominant, then you have to add two of them. The first one will be this one with this guy here. And then the other one will be this guy with this guy here. So you add them together, okay? And it will give you 8% of the time. So if we take a look at this and we do all the possible combinations, you'll end up with these numbers here. So the possibility that two homozygous dominant parents mate is 0.04, etc. You do it for all the different genotypes, okay? When you do that, if you add them together, you'll end up going back to that ratio that we saw earlier, which we calculated from here, right? 0.3 and 0.7. So if you take a look at here, the total big A is 0.4 coming from here, 0.4 coming from here, and then 0.1 coming from here. So these are the offsprings of these guys, okay? And so you can see how, as generations go, if the population is in equilibrium and follows Hardy-Weinberg equilibrium, the frequency of P and Q remain the same. The P and Q remain the same. Does that make sense? Okay. So that's pretty much it for that. Yeah, I'm going a bit too fast, but you can see how yeah, one second. So yeah, then you just do the square root of these numbers that you get, and you'll end up with P by itself, which is 0.3, and then Q, which is 0.7. Okay. Hopefully this is simple, just basic arithmetics. Okay, what if we have 3 alleles? So this is where it starts getting messed up. And then basically you still have the same P and Q, but then you will add another letter for the third allele, right? And so, for example, you can use R here. So the frequency of A will remain as P, frequency of the second allele will remain as Q, and then you have the third one R, and then ST, whatever, until you reach the end, okay? So the difference here is that we have to take into consideration some combinations, because now it's not only a combination between 2 alleles, but it's a combination between 3 alleles. So first, you keep, for the genotypes, you will keep the big A, that's P squared. You will keep the Q squared, which will be the second allele, so A2. And then you'll add a third one, which is R squared, and that will be the third allele in the homozygous state. And then you're keeping also the homozygous, as we've seen before, heterozygous, I mean, between 2 alleles, that's 2PQ, right? And then you're going to add the other combinations that are possible. So the dominant allele with the third allele, the second allele with the third allele, etc. Does this make sense? Easy? Okay. And all this added together gives 1 still. Okay. Yeah?
Like, would there be a question with like 5?
No, no. Yeah, if you get it for 3, you get it for 5, you know. And it increases, no, but what I mean, not like that. What I mean is it increases the chances of you making mistakes if we go to 5 and 6. So keep it low. Yeah.
Like, are these things on the formula sheet or?
The Hardy-Weinberg will be, the rest no. Yeah, so, well, this stuff you can figure it out, right? Just added another letter. Okay, so, what else? So Hardy-Weinberg is achieved. So if you start with a population that is not in equilibrium, after 1 generation, it will become in equilibrium. Okay, because then mating will start being within the same gene pool, and so you'll end up with. Obviously, it has to be random mating. If you have a selective mating, then it will skew the gene pool towards one genotype more than the other. Okay, if every girl is going for tall guys, well, we're gonna have issues there, okay? That's just an example with selective mating, okay? And so, also, this is important to remember that this is only talking about 1 gene. No linkage, because if you start getting into linkage, then that's another story. So only talking about 1 gene, either 2 alleles or multiple alleles. And also remember these how do you call them? Assumptions about the Hardy-Weinberg equilibrium. If the population is not respecting any of these, you cannot apply what we just learned to it. Okay? And this is just a description there that, what do you call it? The actual scientific explanation of this. You can read it or not, but the way I explained it, I think, is sufficient, okay? So, the other thing, the frequency. So when you're looking at gametes and you're looking at the genotypes that they produce, so the frequency of the alleles within those gametes will determine the frequency of the offsprings, okay? And so if you look at the Punnett square that we looked before, and you have the eggs and sperm, then you're going to have the same exact thinking, right? So if the egg is A, sperm is A, then that will be homozygous dominant, p squared, etc. Simple? Yep.
When there's 3 alleles, like, what is it for the dominant?
When there's 3 alleles, what is it for the dominant? So there will always be 1 dominant allele, and the others will be depending on each other. So for example, if you remember the color stuff, there was like the blue with the beetles. I think it was like lecture, right? So there's 1 that's dominant to the other, dominant to the 3rd one, right? Yep. So that's that. And what did I want to say here? Shit, forgot. Okay, hopefully it's gonna come back. But okay, so why do we use this? What's the purpose of calculating this stuff? So it's using by population geneticists to study diseases, how they evolve within a population. A lot of epidemiologists use this. If you want to have your risk factors for getting a child with a genetic disorder, you'll go to genetic counseling and they will do these calculations and figure out what are the chances that your kid will end up with a certain disease. And then it's also important to calculate the heterozygotes in a population because usually they are the carriers of diseases. So you want to make sure that on a higher level in terms of government policies and all that, that you take a look at this. Okay, so there are scientists that are doing this right now to make sure that we avoid problems. I don't know what happened during COVID, but yeah, that's it. So what else? Wait 1 second. Let me just review this stuff here. Oh yeah, so if you see the frequencies being not in equilibrium, so that means there was one of the assumptions that was violated, and you can then use that to determine what's happening to the population. For example, if you see a change in the amount of, let's say, people with sickle cell anemia, then you can imagine that maybe there's a selective force happening right now, including malaria in your country. Usually this is like purely hypothetical, okay? More relevant example is that there is more migration from a country that has too much sickle cell anemia into, let's say, for example, Canada, and that will increase the chance of getting sickle cell anemia, okay? We'll take an example of that, of that in a second, where the African population went to the United States, and they have a higher rate of sickle cell anemia, okay? And you'll see why is that. Chi-square, your best friend. So how do we use chi-square to take a look at our observed numbers? So for example, if you want to test if a population is in Hardy-Weinberg equilibrium statistically, not just going through the p-squared, 2pq, etc., then you would use chi-square. I'll take an example for this genotype here. We don't really care about it. It's just an antigen that's related to the immune system. So there's two alleles for it, either the A or the G. And then you can use the frequencies that are expected, right? So this is your expected frequencies from the 2p-squared, 2pq, q-squared. So these are these guys here. And then you compare it to the observed frequency within the population that you are studying. And then you can see if this population is within a Hardy-Weinberg equilibrium or not. So if you get a list of, let's say you take a sample of 1,000 people, you get their genome, and then you study this allele here, this gene here. You'll get different genotypes, right? Either this one, or this one. So you can count the number of people that have the homozygous dominant, homozygous recessive, and heterozygous genotype. And then based on some expected frequencies that you already have, then you can compare those numbers to the population by first calculating the frequency of this genotype, the observed frequency, and then the observed frequency of this genotype and this one, and then you compare it to the expected, okay? By doing the same exact formula, so observed minus expected squared over expected, and you do the sum of that. In this example here, they're looking at 0.005, so that's the confidence. We still continue with 0.05. Okay, that does not change for us. It's just, I guess they're using a more stringent way. I don't know why they're using them, but there's some sort of scientific explanation behind this. Okay, so we don't really care about the scenario. This is just an example of how to use Hardy-Weinberg chi-square to test a population if it's in equilibrium or not. The statistical way, which is the right way. So that's that. Okay, not bad. So, yep. Oh, yeah. So the degrees of freedom, there is one, because now we're talking about the frequency of alleles, and we have only two alleles. So your class becomes the allele, right? And in this case, we only have two alleles. So that means we have two classes, and then you do that usual -1, which gives you 1 degree of freedom. If we were talking about this situation here, your number of classes would be 3. -1, that's your degree of freedom, so 2. Okay? Thanks for, I forgot that. I think this should have been the first slide there. That's basically what I said. We study genetic variation because it leads to, you know, the phenotypic differences that we have, which is a good thing. And then it gives you the cause of heritable diseases, especially in certain areas in the world where there's inbreeding, and we'll see inbreeding next lecture. And also we can use it to determine the response to drugs, because based on your genotype, you are sometimes resistant to certain drugs. I'll just use a hypothetical example. For example, the Eskimo group that we saw earlier that had more M allele than N, actually 92%. You could, for example, have a treatment that treats sickle cell anemia, and because they have the allele M, that it does not work on them for some sort of interaction in your body, right? So there are examples like this where we use the genotype to see where a drug will be more responsive or not. And if you just go to Europe, that's like they're not far from us, you will see that the medication they have there is much different than here. And there's a reason for it, okay? We don't have the same medication in Algeria that we have in Canada, etc. And it's just because it's adapted to the right populations, okay? And obviously you can trace your ancestors and see where you are from. Cool. So what else? Oh, forgot to remove this. Forget this slide, okay? So now cystic fibrosis. Have you ever seen this? So I was young, I've seen usually it's old people that have this thing here, and I was always wondering what the hell was that. But now I understand it's cystic fibrosis, okay? And it's basically a disease that affects a channel. So, you know, channels, you learn channels in cell bio probably, that take in either nutrients or minerals and all that. So there's a channel that's messed up. And then that channel, because it's messed up, it prevents you from producing mucus. One of the most important parts in your body where you need mucus is your lungs. And so because the mucus blocks your breathing, it decreases the surface area where oxygen goes, then you end up having hard time breathing. And so you need to supplement with this and also use these medications that decrease mucus production, okay? I think there was even a study that was showing that cystic fibrosis, well, I mean, it's clear like this, but to understand it in the scientific way, that it had a higher risk of death related to COVID. And you can see why, right? I mean, logically, it makes sense. You have two problems affecting the lung, right? But if you try to understand related to the cells, you will see that it's related because of to the mucus production, which is also a protective layer for pathogens and all that. So your body uses that to protect you, okay? Why do we care? Because we're studying genetic populations. Population genetics. And so for cystic fibrosis, there is a concept here that I want you to focus on. It's the heterozygous carriers, okay? But let's start from the beginning. So this disease is recessive. That means you need two recessive alleles to end up having the disease. So the genotype equals, so this genotype, the CF genotype, which is equal to Q squared, this is not a 2, it's squared, is equal to 1 over 2,500, okay? So one person in 2,500 individuals has this disease, okay? So if we go from there and we calculate the frequency of this allele, then we do a square root of Q squared, and that gives you 1 over 50. So if there's 1 over 50 people, there's one second, right? So this allele, there's 1 over, let's say for counting in terms of 50 people, there's 1 over 50 the frequency of this allele is 1 over 50. If we look at the healthy allele, that leaves about 49 over 50 that are healthy, right? And so if you take a look at this and try to find the frequency of heterozygous carriers, then you'll do 2pq. And I'll tell you why this is relevant, but just follow for a second. So 2pq, you will basically do 2 times 1 over 50 times 49 over 50. And you can see that there is 1 over 25 that are heterozygous carriers, right? If we have this many carriers, how come we have very little numbers of diseased patients, right? So if there are this many heterozygous carriers, why is it that the disease is rare? And to explain this, you have to think in terms of probabilities. So let's say there are 2 carriers that get kids, and you do your Punnett square. So you have 4 possibilities, right? So then the chance of the child being affected is first the chance that you are a carrier times the chance that the other parent is a carrier times the chance of basically the 4 squares of the Punnett square. So over 4 kids, okay? And so that gives you 1 over 25,000. So you can see how even though you have a big frequency of carriers, it gets diluted because it depends on not just you being a carrier, but another person being a carrier, and then the number of children that you get, okay? What else? So you can do the opposite also. If you have the frequency of people that have the disease, for example, North or European Caucasians, then you can calculate the allele frequency within that population by just playing with the numbers. I've already showed you this before, so you can just go backwards and figure it out. Okay. Yep.
So the 1 out of 4 though, it's like based, that's like the Punnett square, right? It's not like
Exactly.
they have something, this is like Punnett square.
The Punnett square, pretty much, because there are 4 possibilities when you're dealing with 2 alleles, right? If it was 3, then you'd end up with what? Let's not get into it. No, let's not get into it. I don't even want to calculate it. Okay. Sickle cell anemia. This is a disease that is highly prevalent in the US within a certain population. So people with darker skin color in the US are affected by sickle cell anemia, and that's 1 over 400 people. Okay, that's a lot for a disease. Why? As I explained earlier during the beginning of the lecture, probably because of migration of those populations from sub-Saharan Africa to the US. Okay, and there were different migration periods where this happened, so you can review history and see this. But if we want to find the frequency of the allele causing this disease, which is recessive, then we can use this number and then go through the stuff that we just learned, so to find the frequency of both alleles. Okay, so if we have 1 over 400 people that have sickle cell anemia, then we know that those people are homozygous recessive. So that's Q squared equals to this number. Do the square root, and that will give you that Q is 1 over 20. So the frequency of that allele is 20% in the population. No, not 20%. What's 1 over 20? 5%? 5%. And then the rest is the healthy allele, that's 95% of the population. Okay. From there, we calculate the heterozygous carriers because they're the ones that are the most important in terms of disease transmission. And they're interesting for the government and other bodies to follow the epidemiology of a population. So you can use the 2PQ to figure out the percentage of the population that has this, that are carrier for this allele, and that gives you 10% of the population. So there's 10% of this group that are carriers for sickle cell anemia. Okay. Now, why is it that there's a high risk of having this disease? Well, if there's 10% that are carriers, that means this 10%. So if you calculate the chance that the children will have it, you'll multiply these frequencies here. Okay. Yep. The 1 over 4, I'm getting there. Yeah. So if you, so first you find the number of couples. Here we're talking about couples, okay? Not about the children. So the number of couples that are carrying this disease is basically 1 over 10 for one parent, 1 over 10 for the other parent. So that gives you 1 over 100. And then the risk of having an affected child is 1 over 4. So makes sense? This 1 over 4 comes because of the Punnett square, where you're seeing that the homozygous recessive happens 1 over 4. Okay. This is where the 1 over 4 comes from. Okay. So, so if the population is not in Hardy-Weinberg equilibrium, that means it's not respecting one of these assumptions. Right now we're going to look at the selection, force, migration, and mutation. And that should be it, I think, for the lecture. So a good example for this, for a population that seems to not be respecting Hardy-Weinberg equilibrium, is gray seals. So gray seals, they work in this kind of, in groups where they have one dominant male, which usually fights the others to kind of lead the group. So if you see it that way, you would consider that population as being not in equilibrium, right? Because there's one male that is mating with all the other females in the group, and so you would see that the offsprings of that male being kind of half brothers and half sisters, right? And so you would expect them to carry the alleles from that male, right? At least mostly from that male, because he will be the commonality between them. So this structure here is called the harem structure. But in reality, when you look at the population and you analyze their genotypes, you'll see that no, they do not find that. Actually, they have a normal, so they do have the harem structure, but they don't have the half brothers, half sisters. It's mostly full siblings. So it seems like the dominant male is not just making offsprings with everyone. They usually stay in kind of a monogamous partnership, okay? And so this is, I mean, just an example of where you would think that the Hardy-Weinberg equilibrium is actually not followed. But when you go and do the genotyping of these seals, you'll find that actually this is wrong, okay? There's a lot of animals that follow this structure here, for example, lions, etc. And so, yeah, this is that. Yep.
So, like, is there actually one male, or like?
There is one, yes, but in terms of offsprings, they remain within the same partner, right? So it's just who's leading the group, but not necessarily who's having kids with who. Okay, make sense? So if you look at the alleles, you'll find that the offsprings are similar, and so they're not half brothers, okay, or sisters. Okay, now the sickle cell anemia. So the first force that disrupts the Hardy-Weinberg equilibrium is natural selection. And what do I mean by natural selection? So there is a differential survival that happens within populations. If, for example, you are more fit to survive within that population, that you'll be able to pass on your genes, which are more fit for that environment. And so the weaker ones end up getting either dying early or getting killed or whatever by a predator. And so natural selection leads to the better fit genotype to being sustained within generations, and the weaker ones to be removed from those generations, okay? One example of this is sickle cell anemia. So in sub-Saharan Africa, let's look at this one here. Sub-Saharan Africa, there is a high rate of malaria. Malaria is a disease that is carried by mosquitoes. It affects your red blood cells, and it makes them hypoxic, right? And so you end up not being able to breathe, and then you have a lot of problems, okay? You can read on it more, but the purpose here is more the genetics. So these populations, with time, what happened is that, so this is a tropical area, so you can understand the mosquitoes and all that, but these people, with time, they developed sickle cell anemia, and evolutionary biologists think that it's a protective mechanism to malaria. Why? Because in sickle cell anemia, your blood cells are shrunk to the point where they have a very hard time already getting oxygen. But what happens is that malaria, which affects the blood cells, cannot affect them anymore, right? And so it's a better fit, even though both are diseases, it's a better fit to have sickle cell anemia than to have malaria, okay? Because you can live with one, but malaria kills, okay? Both of them kill, but this one on like few days, okay? So that's that. And you can look at the pattern, and it seems to be pretty much the same pattern. There is a few changes, north of Africa and then over here, okay? Also, there's another concept where the frequency of alleles happens in a sort of a gradient, right? So this gradient just means that at this point here, there is less sickle cell anemia than here, okay? And why is this happening here? Probably because malaria here was super strong and here it was much weaker, okay? There were less incidences of malaria in these regions. Yep. Yeah, exactly. there's a lot of forces, not just one thing. But yeah, people will migrate from one spot to the other, and that will cause something called a genetic drift, where you can imagine if all these people here came to the US, then within a few years, the US will be like fully sickle cell anemic kind of, right? And this was shown in the example we had just a few seconds ago, right? Because there was a migration from sub-Saharan Africa to the US, and that explains why that group of the US population has higher incidence of malaria. Cool? Good. I don't know if they're doing it in the government, but they should be doing it. So immigration could be kind of directed by this kind of stuff. If you see that, for example, there's a drift that's going to happen from these regions here and increase the rate of malaria, of sickle cell anemia in your country, then you have to play with those numbers. That's another hot topic. So yeah, that's just a possible idea. Probably they do it, but I'd be surprised. For now, they're busy with other stuff. The other thing is mutations. So as time goes and generations go through, a population that is in equilibrium will kind of become not in equilibrium because of these mutations. And so the more mutations that you have, the more an allele will be, the less frequent an allele will be, because you will be creating more alleles and different variations of that allele. And so with time, with generations, the population will be out of Hardy-Weinberg equilibrium. And this is the example for humans. Okay. With time, what's going to happen is that the allele frequency will reach very low number. Obviously, because new alleles and then different mutations and all that. Question before we go to the next one. Yeah. Yeah, because mutations affect the frequency of alleles. So, for example, let's say you have a lethal, recessive, lethal, recessive, okay, allele. Does that allele happen in a Hardy-Weinberg equilibrium or not? Anybody? No, it doesn't. Because if you get to the recessive phenotype that kills you, then you're removing from that population some alleles, and with time, that allele will disappear. Does that make sense? Okay. Now, those that have mutations in this gene that is recessive lethal will be able to survive, and that's where the natural selection happens, and those will be the more fit to live. If you go to the gym, you'll see natural selection happening right there. The way people are using the machines, man, it's crazy. Yeah, so hopefully nobody's gonna get killed by the machine, but you know, this is how it works. Okay, what do we have left here? A few more slides, I think 3 or 4. Okay, you have these, I don't know what they are, deer populations. Funny how they make these graphs, man. West side, east side, blue, red. The deers are pretty gangster. Okay, so this is another way where there's a mechanism that changes the genotype and gene frequencies in populations. So imagine these two populations as being closed. So this is in Hardy-Weinberg, and forget about the two here. This one is in Hardy-Weinberg equilibrium. And then as they mate randomly, they will keep having the same frequencies. If there's two, for example, 2 deers that come from this side and migrate to the other side, they will bring their own genotype, and this will affect here the allele frequencies, right? The genotype frequencies, and as these start mating with these, it will go back into equilibrium at some point. Okay. I don't know who's calling me, but anyways. What else? There's another mechanism called the founder effect, and this is an example that I gave earlier to answer a question, where there is a group of animals from the same species that travel to another place where there's none of that animal. Okay, so in this case, there is other deers, it's just they're different. So another species or very similar species, or the same species but that has evolved separately. And in this case is where there's a migration from one group, from a place where it is to a place where it does not exist there. Then you will create a new population there. And because of the selective forces in that area, you will evolve differently compared to the parent group. Okay, this is called the founder effect. You probably saw this in 225, if I remember correctly. And so these are mechanisms where the population is no more in equilibrium. Okay, so you have to make sure that these are not happening, and if they are happening, then you cannot use the formula that we learned earlier. Okay, we will learn next lecture how to deal with a population that is not in equilibrium. For example, when you're talking about inbreeding, in that case, you're not really replenishing the. So I'll leave it to the next class. But basically, if you're having inbreeding, you're not respecting what we just learned. Okay, and there's a different way to calculate this. That being said, last question here. Anybody? Okay, so I'll give it to you. It's C, if I remember. Yeah, it's C. And so it's saying the Hardy-Weinberg principle tells us that in a randomly, this is important here, mating population, in the absence of all these selection mechanisms, the frequencies can be predicted from, the genotypic frequencies can be predicted from the allelic frequencies.